Good morning, AP Biology. Happy Tuesday and happy Earth Week. Uh, Earth Day week. I don't know how to say it, but anyway. So first off, it is Tuesday. So here's a little trivia for you. Since we are going to be talking a little bit about GMOs, since it's kind of related to this prokaryotic gene control, excuse me, I have some GMO questions. So, first GMO ever made, what year? What do you think? Actually, fairly early, 1973. It was a bacteria that they engineered to be resistant to an antibiotic. Obviously, we did that just to kind of prove we could. I mean, why would we want to make antibacterial resistant bacteria? That's not a good idea. It took a while before we could figure it out to do more advanced organisms, but we have obviously. And the first food product was put on the market in 1994, the first food GMO. What food was it? Post in the comments what you think it was. All right. And now, actually, that food is still not really on the market, but a lot of GMOs, mostly in like soy, corn, and commodity crops, and we mostly insert genes to be resistant to Roundup. So we can spray the whole field and it kills everything except for the food. All right. So today I am going to put a document on your assignments in the classroom. Okay, so it's going to look something like this. Okay, and it's we're going to talk a little bit about basically making a genetically modified organism. We're going to transform a bacteria, add a new gene to be expressed in this paper model. So you can kind of see how it's done in bacteria. All right, so a little vocab that you will need to understand for this activity. First off, we talked about bacterial having one circular chromosome, which is true. However, there is a little exception. Sometimes it gets these other little circular chunks of chromosomes off to the side that are really tiny. And we call these plasmids. They are small, circular, self-replicating DNA. And the advantage of this is that bacteria can actually copy these and pass them on to another bacterial cell. So they don't actually have to inherit all their DNA from their parent cell. They can if some bacteria gets antibacterial resistance in a plasmid DNA, it can just copy that and like pass it on to the bacteria next to it, which is kind of crazy. So we can spread that really easily. All right, so plasmid DNA is some of the easiest DNA to get bacteria to take up extra DNA in. Okay, and so how we do that is we use restriction enzymes. If you remember that from the DNA lab that we didn't quite get to do, a restriction enzyme is an enzyme that will cut the DNA. Okay, and it can cut in two different ways. It can cut flat across the double helix, and that's called a blunt end. And that's not as useful as cutting so that the bacteria has sticky ends. And when it has sticky ends, it means you have your double helix here, and it has the site where the restriction enzyme recognizes. It will be some sort of repeating pattern, like maybe A, T, T, A, A, C or something. And then it cuts down on one side, across the double helix, and down on the other side. So when it breaks apart, it has maybe 4 or 5 bases that are exposed, okay? And that pattern of bases will want to match up with its complementary pair and either reanneal where it was cut, or if there's another piece of something that was cut in the same pattern, it can attach to those. Okay, and so what we're going to simulate with this little paper activity is the transformation of DNA, the transformation of bacteria, altering the genotype and phenotype by inserting DNA. Let's see. I think the particular one we are inserting here is an antibacterial resistant gene. I think as you go through the reading, you'll see. No. It's a GLO gene. Okay. GLO genes are really commonly used to identify if we had successful transformation because you can actually physically see it. Okay, so think back to our operon idea when we were doing the notes. Okay, so let's say we have this bacterial operon that, let's say it has the lac operon. Let's say that's what it is. Okay, well, we know we can turn on that gene if in the presence of lactose. So let's then further say, all right, we're going to insert this gene at the end of the lac operon, this GLO gene. So as long as there's a site that we can cut it there, we can insert the GLO gene. Okay, and maybe we have another gene as well, like we're inserting the GLO gene with antibacterial resistance or something like that. And now when it reanneals, it's all in that operon. Okay, and so we can put that bacteria in the presence of lactose, and then in the presence of lactose, it will turn on that operon, and your gene should then glow as well. And if it glows, you know that the antibacterial resistant gene is inserted as well. Okay, so lots of times the GLO gene in actual microbiology is used as an indicator that some other gene got inserted as well. That is a little harder to see. Okay, so you are going to be doing the GLO gene from jellyfish and inserting that into a bacterial plasmid in this activity. The directions are very well written out, I think. You just follow along. Okay, so along with the directions, you will get a plasmid DNA section. It's just this one. You'll cut it and tape it in a circle, and that's your plasmid DNA. It tells you that in the instructions. Okay, then it will tell you which restriction enzyme you're going to use, so you'll know where to cut. All right, and then you will have the GLO gene as well. Okay, this is going to be a linear gene because it comes from eukaryotes, so you can just lay it flat. Okay, I can't scan this in color, so when you get the green paper where it will say print on green paper, yours will probably be white. Just take a colored pencil or a marker or highlighter and just highlight that green, okay? Okay, you're gonna cut this with the same restriction enzyme, right? Because then the sticky ends will match up. And the green GLO gene will be able to be inserted into your plasmid DNA. Okay, again, the directions are pretty straightforward, but read them carefully so you understand the process. And then there's some questions on it. Okay, by Thursday, I would like you to finish this and finish the questions, scan those to me. And I want a picture of you wearing your plasma DNA with the GLO gene inserted on your head like a hat. Okay. All right. So look for this attachment to assignments very soon. All right. Have a great day. Bye.